Hey, good morning, everyone. Welcome to COVID Impact 360 Conversations. My name is Larry Cole. I'm your host. Very pleased to have an old acquaintance of mine on today. We met about 3 years ago when long COVID was starting to become a thing. It's Dr. Bruce Patterson. He's CEO and founder of HealthBioAI and InCell DX. Good morning, Bruce. Good morning.
Morning, Larry. Nice to see you again.
Yeah, it's great. So I like to start where I think is a logical place to start. People don't, or at least people that might be watching this may not know your background, and they won't realize how hard I've tried to get you onto this show. And there's a very specific reason why it's been hard to get you on this show, because you're busy. But if I could get some of how you got to pre-pandemic, let's move up to that point there.
Right. You know what? It's really an interesting story. And someday I'll probably write a book about it. But this is actually my fourth pandemic. I'm a viral pathologist. I started HIV research in 1990, if not a little bit before, when I started working on RNA viruses. And of course, everybody working on RNA viruses was pulled into HIV and the RNA retroviruses. So, it started there. And, my major area of focus was viral reservoirs and, and pathogenesis. So, once we went through that, we went through H1N1 and several other pandemics. And, I happened to be in China in January 2020 in Shanghai, talking to a cancer company about a new immune profiling test that we had developed. And then the next day, I was supposed to go to Wuhan to a company there that had just validated our lung cancer assay. And I remember getting an email that night from my colleague in Wuhan saying, "Don't come. You can't come. The city is shut down. I can't go into much detail, but you won't be able to get into the city nor get to our laboratory." So I thought that was very curious. And the next day I went to, the cancer company in Shanghai, say, what is going on? And they showed me an early report in Chinese, of course, of some of the laboratory findings on the first few patients that we now know to have had SARS-CoV-2. And I was struck, obviously being a virologist, in the right place, in the right time, looking at data, which is what I do, what I've always done, and seeing this really profound innate immune response with numbers of TNF-alpha and CCL5 antis and interleukin-6 that were off the charts. Interleukin-6 is in the thousands per picoliter of blood, and when it should be 1. So that day, it was also highly suggested that I go back to the United States. There was a television series, I can't remember what it was, but it had something similar where this reporter was stuck in China and, got involved in this mass exodus. Well, I wouldn't say it was a mass exodus, but nevertheless, I immediately got out of Shanghai and got back to the West Coast and started thinking about what this virus could be. And then, of course, the reports started coming out, and obviously it was an emerging infection because the first thing you noticed was this innate immune response, which is all monocytes, macrophages. That's really all you have before adaptive immunity kicks in. So it was truly a monocyte macrophage disease, which can be damaging in terms of what's being produced, but it was something that really I hadn't seen before, but nevertheless felt prepared to try and modulate the immune system so that we could temper what was later called the cytokine storm. So started for me. And by February 2020, I was already doing clinical trials with some drugs for acute COVID. And we followed those patients for 30 days, 60 days, 90 days. And, at 90 days, most patients were not dead. Most patients were better, but by no stretch of the imagination was their immune system normal. And then that's when we applied machine learning and AI to that data and showed that it was abnormal, but it was abnormal in a way that was very, very different than acute COVID. So really, that was when we first came upon long COVID, was really the summer of 2020. And of course, that led to our first paper where we developed the algorithm with long hauler index and severity score to really describe the different phases of COVID, if you will, both acute mild, acute severe, and long COVID.
Well, as we were talking before the show, I think now my background's business, and I understand risk, I understand investment and all of that, but I would hazard to guess that very few people really understand what it means to run a drug development or a therapy development company. And I say that out of respect because we hear words like big pharma and all the rest of it, but believe me, people like big pharma when they have cancer and they're getting drugs. So it really is a perspective conversation, but I fought to get you on my show because as a virologist, you have a unique view, but you're also in a development company. And I want people to get a window into that world. What does it mean to have that kind of risk? If I could get you to kind of frame a conversation around when you start a company like this, the stuff it takes to run a company like that, because it is not easy. I've known you since just after this long COVID term came out. It was long hauler and then it became long COVID. And that's when you and I first talked on the phone. But you've been down a lot of paths since, and that's like 3 years now. But if you could kind of address that, Bruce, I'd really appreciate it.
Yeah, that's another interesting facet. And really it was when I was directing virology at Stanford that, I was at the time I was in an environment, that was all about business. It was Silicon Valley. So it was how do you take something from discovery at the bench all the way through commercialization? And that is probably one of the hardest tasks in the world. Monetizing science is extremely difficult. And frankly, it's driven by what I call the 2 R's, reproducibility and robustness. Because I'll tell you, every researcher who discovers something on the bench, their first call is to their colleagues. Their second call is to venture capital, right? And before you even know if it's robust enough to make it as a product. And so, yeah, there's tremendous risk early on. And the only thing that mitigates that risk is the 2 R's, making sure that whatever you've discovered is reproducible and making sure that what you discovered is robust. And sometimes it is, and sometimes it's not. Well, the ones that it's not, you certainly don't pursue in doing product development because it's costly. You're absolutely right, Larry. I mean, it costs a fortune. I mean, if you look at the numbers from taking, say a drug from discovering a compound to through phase 3 or even phase 4, you're talking, probably several $100 million to $500 million per drug. And, having lived through it, that's not a number I want to have to go out and raise. But the bottom line is when you really find something and it works, it's time to go. And that's when I I left Stanford prior to, obviously, way prior to the COVID crisis. We started IncelDX, which is a precision diagnostics company. Did product development, FDA submissions, CE-IVD stuff. We learned a lot about regulatory, and regulatory is all about, reproducibility and robustness. And the other thing, unlike academia, so academia, there's always a question about how robust data and publications are. Well, there's nothing that makes it real and robust than taking it through agency, because that's when you really know you have something, is when it stands up to the rigors of the regulatory environment, especially in the United States. That's part of the equation. And what we did is we developed precision diagnostics. Then, most importantly, this middle piece that we don't really talk about that much is data analytics. And we started doing that in probably 2016, 2018. And we had our first diagnostic for bladder cancer that was based on machine learning. And so that was well before AI became part of the vernacular like it is right now. But that's essentially what we were doing. And it allows unbelievable analytic power. Because, for instance, if you take next-gen sequencing and you can detect 100 genes, that means you have 100 parameters on a sample that you can give to some bioinformatics person. But if you can detect 20 different biomarkers on an intact cell, you have 20 markers in various combinations. That's 20 factorial. So you're talking billions of combinations. And the panel that we have right now, that we just obviously published the long COVID diagnostic algorithm and the chronic Lyme algorithm in Nature Scientific Reports a couple of weeks ago. 14 biomarkers in various combinations is 89 billion different combinations of those 14 biomarkers. Okay.
Yeah. Crazy number.
When people say, hey, Bruce, why don't you add, this marker to your panel? Why don't you add that marker to your panel? They're all great suggestions. And it's what you're alluding to, Larry, that makes that difficult. Like, we've already developed this product. It's gone through our product development process. We've spent a ton of money to get to these 14, which was, we helped obviously by machine learning and AI. To add another marker seems easy, and it's easy on the lab bench, right? I know that from my academic career. But in terms of doing that, in terms of a product, you're talking about, 6 to 12 months of work and, significant expense to add one marker, right? And so, that's how we think. And we think our markers, with the help of, machine learning, really represent a cross-section of all the different arms of the immune system. And so we're really happy with our markers. And like I said, we published the long COVID diagnostic algorithm, the Lyme diagnostic algorithm. We're working on ME-CFS, which next month I'll be presenting, or I'll be presenting that in the webinars coming up in October, and then next month at the ILADS meeting. But the long and short of it is, we think ME-CFS may be 5 different diseases. There's 5 clear immunotypes, so they're completely separate under the heading of ME-CFS. So we're starting to decode ME-CFS, which is very heterogeneous and different from long COVID and chronic Lyme, of course. But the bottom line is, this tool gives us the ability to take a look at that. But it's, like you said, significant product development teams, regulatory teams, bioinformatics teams. It takes time and it's costly.
I don't think the majority of the public understand, and it's not their fault. They're not scientists and they're not developers, but I don't think they understand that COVID and long COVID we're in the first minute of a 24-hour clock. Like, this is not something that's been studied for the last 25, 30, 40 years. This is so new. So when I try to publish as often as I can find it, anytime that you're doing company announcements, only to try to keep the public, because the public doesn't know that this is actually going on, or they don't, or they'll say a statement like there's nothing being done when the opposite is true. There are
Oh yeah,
people feverishly working on this right now, feverishly.
absolutely. And you know, it gets misinterpreted like, oh, well, Dr. Patterson hasn't been in Twitter for months. Is he just backing down from,
Yeah.
his hypothesis? I'm like, no. You know what? I have a phase 3 clinical trial that, we've been working 12 hours a day, 7 days a week for the last 6 months to push that go button, and on top of that, raising money to fund the trial. You know what? It's interesting when, as you say, people think that they know what you do, but at the end of the day, when you get into product development and especially taking a drug through the FDA, we were over the moon that the FDA approved our trial design in 28 days.
That's
For
probably
me,
pressure on them, though. It has to be pressure on them.
yeah, you know what?
Uh,
But I'll come right out and I say, the interactions with the agency for me were fantastic. I know it's probably rarely said, but you know what? If you respect their input, you address their input, everyone put their egos aside and say, hey, can
yeah.
we meet in the middle somewhere? You know what? You can do what you need to do. And at the end of the day, they're basically saying, this is what you need to do. And, from my perspective, being in the business for 25 years, if you just say, yes, that's a great idea, I will do what you told me I need to do, and hopefully we'll achieve our desired outcome. I think it's when you push back and say, no, I want to do this, that, you get into contentious discussions. But, at the end of the day, they kind of lay out a roadmap and you say, okay, I'll take that roadmap. And I think if we do this and we do it the way you outlined, and we get to the end, we're going to have the outcome that we want. And I think that's been my experience with the agency. And again, that's why I'm so thrilled to be in the position that we're in on the precipice of starting our CT trial and, hopefully getting our drug combination approved.
That most people don't realize that when you decide way back when you're a young man, okay, I'm going to go through this career path of science, that they didn't say that you're also going to be somebody that would have to be very well spoken, be able to lead a company, be able to talk on a podcast. This is so far left field of what your official training was at the time. Be able to raise money, be able to talk to investors in a way that they understand, be able to talk to science like the FDA, being able to talk to all these various stakeholders in a language that they understand. And I can guarantee you that training didn't come from a class anywhere. That is just pure experience.
You know what? It is. And I think, as I tell students, I told my own daughter, as they're going through their academic careers, whatever career path they're doing, and they're studying in books and looking up articles and this, that, and the other thing. And they think that, getting A's in every single class is going to be the key to their success. I think the one thing I tell them, and I told my daughter this, is it's not so much what information you have, it's your ability to communicate it.
100%.
If you can't
100%.
communicate, if you
Yeah.
can't communicate your discoveries, if you can't communicate the underlying mechanisms, if you can't communicate what you're working on and why, then how effective are you? And frankly, you know, you could be super intelligent and not be able to communicate. And conversely, you could, by, standards, not be that intelligent, but be able to communicate your ideas effectively. And, at the end of the day, it's a combination of both. And, I think being able to communicate what you do, what it means, is there's nothing more important in life than that.
Well, you just hit a lot of areas that are extremely important to me. I think being understood. So I'm going to back up a bit. When I'm addressing groups, I literally try to speak between grade 7 and grade 10. And I don't, because I don't want to talk over somebody, nor do I want to talk under somebody. I just want to be understood in the language that they walk away. I get. I've been involved in too many meetings where somebody won't ask me a good question because they're not quite following it and their ego gets in the way. So they won't ask the question. I want to get the questions. So, and I'm bringing this up because if we take a snapshot of your company today, right now, and I know what's coming up because we're going to talk about it in a moment, but where are you at right this moment in time with your developments?
So it's such a great question, and I think we can kind of roll in, the IncellDX HealthBioAI
Yes.
kind of conversation, and frankly, IncellDX has been diagnostics, precision diagnostics, and always has been. That's the DNA of IncellDX. HealthBioAI is a much larger entity that includes, pharmaceutical patents or pharmaceutical pathways or approvals or our pipeline. But what brings them together, which is glorious, is this data analytics portal that our team developed that takes all the incoming data from thousands and thousands and thousands of patients, takes all the data from our R&D, takes all the data from we have a wearable to input data. All of that goes into a singular data analytics portal. We use this portal not only to develop new algorithms for new diagnostics. We may stay with the same panel and have 8 different diagnostic algorithms for God knows what. Certainly now, along COVID, Lyme, and next up is probably ME-CFS. But most importantly, that data analytics portal informs us as to our drug discovery. Where do we go next? What's next in our pipeline? And we have that pretty much laid out. Next month, I'll be presenting ILADS, the national Lyme meetings, on an enormous discovery that we found just 2 weeks ago. Very similar to what we found in long COVID, but something that will address the chronicity of Lyme disease, which, I wrote an op-ed on, how Lyme is the pandemic that COVID uncovered. And it's true. I mean, we have uncovered so many patients who had undiagnosed or inadequately treated Lyme because acute COVID causes immunosuppression. That is, I think, the most important point about acute COVID and long COVID that is absolutely missed. Acute COVID causes sometimes profound immunosuppression. At its best, you have decreased CD8 counts. At its worst, you have decreased CD8 counts in the single digits, means CD8 percentages in the single digits when it should be 30%. And those cells are exhausted and express exhaustion, T-cell exhaustion markers. And in our first paper in 2020, they had downregulation of granzyme A, which of course is necessary for CD8 killing of virally infected cells and tumor cells for that matter. Acute COVID causes immunosuppression, however mild, okay? And so to me, when somebody says, oh, it's just a cold, it's by no stretch of the imagination, just a cold.
Yeah.
With all the effects of
I dismiss that stuff. I just dismiss it outright.
The pathology that we and others have published on, the spike protein and what it does for ongoing activation through antigen presentation by monocytes and vascular inflammation. I'm telling you, it is not a cold. And it's out there, and we're getting additional surges. So, you know what? COVID, I've heard from a number of investors, oh, we're not going to invest because we don't believe, COVID is going to be, lucrative or part of the future. Well, number one, we're a chronic inflammation company. Okay. just so happens that long COVID is a huge unmet need and our first indication, but so is chronic Lyme, so is chronic fatigue syndrome, so is fibromyalgia. I mean, we have a great pipeline for both our diagnostics and our therapeutics that we will be rolling out in the coming years. But this is not a COVID company. And like I said, HealthBioAI represents the culmination of bringing in our precision diagnostics with a data analytics portal for drug discovery, and then, of course, our pharmaceutical platform.
So you said something about investors there for a moment, and I'm kind of baffled by that. And I'll put it to you this way, and these are all very rough numbers, but I post about 100 times a day about this from everybody, so I get a sense of numbers. So I believe the best thing to happen to the ME-CFS world and the Lyme and the fibro and anything that deals with immune, I think the best thing that ever happened to those worlds in a very positive way was COVID, because it brought a lot of attention to those, and attention that they weren't getting. They felt like they were in tiny little silos and they weren't getting heard. All of a sudden you've got 300, 400, 500 million people with some type of a post-viral infection. So this goes back to my investment. My investment thesis is that there probably is no opportunity in the world, period, in biomedicine than COVID. There is no greater opportunity to be in, if you're in with the right company, doing the right things and trying to do the right therapy. So yes, I know biomedicine is a risky business, but you're dealing with an awful lot of people, which people spells opportunity, and that's global. It's not like it's like that's going away.
Yeah, no doubt, Larry. And you hit the nail on the head. I mean, 20 to 30 million Americans with long COVID, 1 in 15 by the CDC's count, way more
Huge.
worldwide. The TAM, our total addressable market, is enormous, right? But it's such a conundrum because it has been an absolute slog raising money. I mean, we could have started our trial in February if we had the funding to do it. You know, we didn't expect to get a, approval to proceed from the FDA so quickly.
Yeah.
And so we had to scramble to raise money, at least get the trial site set up. But you know what? I am aghast, and people I talk to are aghast that, raising money for what we're doing hasn't been just, smooth sailing. And, I like I said, the best description in my mind is it's been a slog. So
well, it isn't for the weak of heart to be in this business to begin with. And then, we're in a business where we barely defined what COVID is. We barely defined what long COVID is. Testing is still a bear. It's early days, but see, when I say that in my head, that spells opportunity, but that's just the way I'm hardwired. So I've used up a lot of your time, and I know that you don't have that kind of time. I think what I want to kind of finish up on is that you've got 2 webinars coming up, one on October 1st, one on October 9th. I think one of them is for physicians and one of them is for patients. I'm going to leave the link on this when it gets posted on my YouTube channel. But do you want to speak to those? Like what you're going to be?
Absolutely. And I think it's a great opportunity with our partners in the UK, Peplo Bio, to really go through what just came out in scientific reports we just published. But most importantly, this test panel that we do shouldn't be a mystery. There are certain and we can do a better job, I think, on the report, and we're already talking about it, to make it more like a Boston Health report, Boston Heart report, where, it's nicely color coded and there's out of the 14, maybe there's, the 3 that define vascular inflammation, my 3 bullet points for is answering the question, is this Lyme? All of those things need to come out. And, like I said, we've been so busy, we haven't had a chance to make a really great customized report that's educational. And in lieu of that, I think we're doing these 2 webinars, both for physicians and for patients. where we explain every single biomarker, why we have it, what does it mean if it's elevated or not elevated, but most importantly, going through our algorithms, because so often, and this is another thing, patients will put their reports on Twitter and they'll say, well, this is up and this is down. Well, 6 out of the 14 are up, and no, it doesn't work like that. But that's on me to explain, our algorithms and how it takes, 2, 3 of the values and puts them in a ratio or puts them in some formula that informs us with much greater clarity than any one cytokine or chemokine on its own. In fact, there's very little value. I wouldn't say little value, but there's less value looking at individual cytokines. So for instance, if a patient has a report and I'm seeing them during telemedicine and their interleukin 6 is, 100 instead of 1, and I see elevated TNF alpha, tumor necrosis factor alpha, that's the hallmark of acute COVID. I'm like, you just had acute COVID in the last 1 to 3 months, okay? Because your markers of, acute COVID, IL-6, TNF alpha, are elevated, but there's a lot of information in those reports, and I think this is all part of us educating and disseminating all the great information that's contained with those, because we need to get away from. There was a great article on JAMA that people really need to realize. That article said none of the routine diagnostics
I
are
posted it.
diagnostic of long COVID. Okay. So you know what? You're not going to get to where you need to go looking at CRP, sed rate, D-dimer. All of those are so non-specific that they're almost inconsequential in following these patients with chronic inflammation. You really need to get down to specific inflammatory patterns to, number one, diagnose patients or say what they have, and 2, monitor them, see that the long hauler index goes down to normal after treatment, things like that. I think it's absolutely critical. And that's the other thing. I don't know how people can even try to treat long COVID if they don't even know that their patient has long COVID, right?
There's the dichotomy.
I see all these papers about this, that, and the other thing. First thing I ask is, okay, how'd you rule out that they had Lyme? How did you rule out that they didn't have Epstein-Barr or cytomegalovirus reactivation, which, again, it's all being lumped together by this symptom complex of fatigue, brain fog, and post-exertional malaise. I'm like, come on. There's at least 5 inflammatory conditions, chronic inflammatory conditions, that have that symptom complex. So, and then they address it by saying, oh, there's all these phenotypes of long COVID, and some have these symptoms. No, you know what? For me, there's very few, if any, symptoms that you can't trace back to inflammation. Take anxiety and depression. There was a study that came out, oh, patients with long COVID have low serotonin, we should give them Prozac, right? Well, what's notorious for causing low serotonin? Interleukin 1 beta and TNF alpha. Okay, get rid of the inflammation, then see where their anxiety is. And having treated a lot of these patients, you get rid of the chronic inflammation. All of a sudden, anxiety gets better, the POTS goes away. The one thing very few people ask of long COVID patients, are you cold when you shouldn't be cold? And are you hot when you shouldn't be? It's temperature dysregulation. That's my window into their blood vessels. I know they have blood vessel inflammation because they can't regulate their body temperature and they can't regulate their blood pressure, and they have POTS, right? So, you know what? Some of this is just being a pathologist. I'm looking at pathways. I'm not looking to put a Band-Aid on, symptoms. Oh, I'm going to give them metoprolol or propranolol because they have a, racing heartbeat. Well, yeah, they have a racing heartbeat because their blood vessels are inflamed and they don't have control over contraction and dilatation. So, you know what? Control the inflammation, that takes care of itself. So we have to get away from this concept of treating all these different symptom complexes that have been published. Are they real? Absolutely. But if you really look deep enough, the overwhelming majority are related to chronic inflammation.
Boy, there was another hour of talking just in what you just talked about, and we don't have that time. But I'd like to end on this final thoughts, because there's two things that I fight heavily on. Is the vast majority of my followers are doctors and scientists and researchers by a factor of 9.9 to 10. I mean, it's that many. But so information is a big deal. I'd like to know more about where, at the end of the day, do you want to see yourself, say, even 1 year from now? Is there something coming out 1 year from now? Just if you could, or is that just an unfair question?
No, you know what? How can you not think about that? And I would say that based on what we just discovered, I think a year from now, we're going to be talking about something really exciting that may transcend chronic inflammation, autoimmunity, etc. And I'm really excited to publish it and really excited to talk about it. So,
well, I seriously can't thank you enough for 40 minutes on your usual 12 to 16-hour days. That's a lot of time out of your day. So, Bruce, thank you so much.
My pleasure.
I appreciate.
Yeah. Thank you for the opportunity.
It's all good. So you take care, and we'll talk to you again soon.
Thank you very much.